Hi, my name is Kshitij Dwivedi and today I am going to describe KIHI 2014 dataset which contains human fMRI and MEG responses to images. Please scan this QR code to read the paper and learn more details. The dataset consists of 92 images with superordinate, ordinate and subordinate categories for which the fMRI and MEG responses were recorded. In the fMRI experiment, there were 15 subjects who performed 2 sessions each. The task was to detect fixation color cross color change. In the MEG experiment, there were 16 subjects who performed 2 sessions each, and the task was to detect a paperclip image. The data provided here is in the form of representational dissimilarity matrices, also known as RDMs. In fMRI, we provide ROI RDMs, and in MEG, we provide RDM for each time point. The stimulus set consists of 92 images that are divided into animate and inanimate categories. The inanimate category is further subdivided into natural and artificial images. The animate categories is divided into humans and non-humans, which are further divided into body and face. Here we show an example image for each category. Now let's look at how MEG RDMs are calculated. For a given pair of images, corresponding MEG response, calculate the distance between the MEG response for this pair of images and use it to populate a matrix that has dimension stimulus cross stimulus. And if we calculate all pairwise comparison, we can use it to fill the matrix, and this matrix is known as representational dissimilarity matrix, or RDM in short. For MEG, we provide the RDM at each time point from -100 millisecond to 1000 milliseconds after the stimulus onset. Similarly, in the case of fMRI, a given pair of images and the corresponding fMRI response for a given region, we calculate the pairwise distance between the fMRI responses and use it to fill the RDM. And if we calculate all pairwise distances for the stimulus set, then we get a stimulus cross stimulus matrix for fMRI RDMs. Now let's look at how to load the data using Colab. So this is the Colab notebook that describes the data set. So first we run this cell to import the libraries, then download the data set. And there is a code to load MEG RDM and visualize the RDM at different time points. So here you can drag the slider to visualize the RDM at different time points. Now we can load the fMRI RDM from early visual cortex and IT and do some example analysis. We have provided an example for fMRI comparison with MEG, MEG comparison with the computer vision model, and fMRI comparison with the DNA example. So let's look into more detail about fMRI MEG example. So what this will tell us is when does the representation of a given ROI, let's say IT, becomes more similar to MEG RDM. So when we compare the RDMs of IT and EVC with MEG, what we find is MEG RDMs shows higher correlation with IT at a different time point, suggesting that this representation is processed later in the brain than early EVC representation. Thank you and have fun.